All right, welcome to week 2 of Anatomy and Physiology. We're not really talking about anatomy today, mostly physiology, and the level of physiology that we'll be talking about is at the chemical level. But these are important things you need to know, or you should be aware of, I guess, to help you through the rest of the year. So we're going to get a quick chemistry course. So, when we talk about physiology, we're really in the realm of biochemistry. That's the study of molecules that compose living organisms and work in living organisms. These things are like the carbohydrates, fats, proteins, and nucleic acids. And understanding what each of those polymers does for the cell structure is going to give you a good head start on basic physiology. You might understand more about nutrition, and they do certainly have a role in health. But we got to start at the chemical level. So a chemical element is the simplest form of matter that has unique chemical properties. It's identified by an atomic number, which is the number of protons in the nucleus. They are organized on the periodic table of elements. They either have a 1 or 2 letter symbol. And if they have a 2-letter symbol, the second letter is always lowercase, the first letter uppercase. And their number is also on there. There are 91 elements that occur in nature. 24 play roles in humans. However, only 6 are really, really abundant. Those 6 make up 98.5% of the body weight. Those 6 are oxygen, carbon, hydrogen, nitrogen, calcium, and phosphorus. The others are trace elements, but those trace elements still play a major vital role. Some are minerals, they are inorganic elements, they're extracted from the soil by plants and passed up the food chain to humans. They make about 4% of the body weight, mostly calcium and phosphorus. And they also provide for body structures such as bone and teeth, enzyme functions, nerve cell, muscle cell functions. So the term atom was coined by a Greek philosopher. It means indivisible, the smallest unit of matter, was first explained using a planetary model of atomic structure in about 1913 by Niels Bohr. It's useful as a schematic. It will help you understand the basic chemistry that we'll be talking about today. It's not entirely accurate. It doesn't fit all the experimental evidence, but it does work. So we have a nucleus in the center of that atom. The nucleus contains the protons and the neutrons, protons with a positive charge and neutrons with no charge. Protons and neutrons are approximately the same size, which is 1 atomic mass unit. And around that nucleus are variably referred to as shells, orbits, or clouds. Now, in the planetary model, they were called orbits, and they were distinct, and you could have 2 electrons in the first shell, 8 electrons in the second, and so on. Then they began to be thinking of them more as shells, and now we know experimentally they really exist more as clouds, probability clouds. But thinking of them using the planetary model is a good way to get started in understanding chemical reactions, the kinds of things that we need to know at this level for an anatomy and physiology course. Here is a more accurate depiction of what the current model of the nucleus is, and you have electron clouds and nodes in between those clouds, and even those clouds are just probabilities. They're not we can get into it very deeply, but I don't think we really need to for the purposes of understanding the basic biochemistry of living organisms. One of the things you talk about when you talk about elements are isotopes. They are varieties of an element that's different only in the number of neutrons. And so from a practical standpoint, all that does is change the mass of that element. Those extra neutrons will increase the atomic weight, and isotopes of an element are very chemically similar because it's not the nucleus that controls how they combine with one another. It's the electrons, and specifically the electrons in the outside orbit known as the valence orbit. The atomic weight or atomic mass of an element, it accounts for the fact that the element in total is a mixture of isotopes. So that's why you don't have whole numbers for most of the atomic masses. Hydrogen, for example, has 3 common isotopes. Hydrogen itself with 1 proton, 1 electron, no neutrons. Deuterium with 2 neutrons, and deuterium with 1 neutron, and tritium with 2 neutrons. The other important thing about isotopes is isotopes actually tend to be radioisotopes. They are unstable and they decay and give off radiation. They call it radioactivity. Every element has at least 1 radioisotope. Their intense radiation can be ionizing, which would be called ionizing radiation. It can eject electrons, destroy molecules, create free radicals, and cause genetic mutations in cancer. For example, ultraviolet radiation, X-ray, alpha particles, beta particles, and gamma rays. So when we talk about radioisotopes, we have to talk about their physical half-life. That's the time required for 50% of that radioisotope to become stable, to decay. And then we also have a biological half-life of radioisotopes, which is the time required for 50% of it to disappear from the body by whatever means. Standard measurement for radiation dosage is the sievert. More than 5 sieverts is usually fatal. Standard acceptable exposure is 50 millisieverts per year. That exposure comes from background radiation. There are natural sources, radon gas, cosmic rays, and we average about 2.4 millisieverts per year. Artificial sources also are out there, X-rays, color TVs, so on, and that would account for about 0.6 millisieverts per year. First woman to receive a Nobel Prize was Marie Curie, and she was also the first woman to receive a PhD. She came up with the term radioactivity, discovered a couple of radioactive isotopes, polonium and radium. She trained physicians in the use of X-rays, pioneered radiation treatment as a cancer therapy. Unfortunately, all of the dangers of radiation were not known at the time, and Madame Curie died of radiation poison at the age of 67. Another different form of an element is an ion. An ion is a charged particle. It could be an atom or it could be a molecule. We'll see both as we go through the year. And the reason it has a charge is it has an unequal number of protons and electrons. So when an ionization occurs, it's the transfer of an electron from one atom to another. That will create an anion, the particle that has a net negative charge because it has gained the electron, and a cation, a particle that has a net positive charge because it has lost electrons. And as you know, positive and negative charges are attracted to each other, so ions with opposite charges are attracted to each other. So if we have a sodium atom with 1 electron in its outside orbit, it could, if it gets rid of that 1 electron, it will leave it with a second orbit being its outside orbit, so it will have a full outside orbit. And if chlorine picks up that electron, chlorine will now have 8 electrons in its outside orbit, and it will have a full outside orbit. So if sodium ionizes with chlorine, we end up with sodium chloride, table salt. Sodium has 1 less electron, chlorine has 1 more electron, but their outside orbits are stable. Ions generally give us salts. Salts are electrically neutral compounds of cations and anions, but they are readily dissociated in water, so they act as electrolytes. So table salt is a salt. When you put it in water, it separates into sodium ions and chlorine ions. Same is true with calcium chloride. Those electrolytes, things that ionize in water, form solutions, are capable of conducting electric currents. So we use electrolytes for chemical reactivity, osmotic effects, electrical excitability of nerve and muscles, and the electrolyte balance is one of the most important considerations in patient care. Imbalances in electrolytes can lead to coma or cardiac arrest. Free radicals are unstable, highly reactive particles with an unusual number of electrons. They're produced by normal metabolic reactions. Radiation can do it, some chemicals can do it. And when they are present, they trigger reactions that destroy molecules. They can cause cancer or the death of heart tissue and probably have a role in aging. Superoxide is a very common example of a free radical. We also have antioxidants. Those are chemicals that neutralize free radicals. So there's an enzyme called superoxide dismutase, or SOD, that converts superoxide into oxygen and hydrogen peroxide. Selenium, vitamin E, vitamin C, carotenoids are antioxidants that can be obtained through the diet. So those atoms combine together to give us molecules. Molecules are a particle that's composed of two or more atoms held together by a chemical bond. Now, there are lots of books with very specific definitions about the difference between a compound and a molecule. So your text is going to call a compound a molecule composed of two or more different elements. It's represented with a molecular formula, identifies the elements and how many of each element is involved. It could also be identified by a structural formula, which will give you an idea of the actual location of each atom. Isomers, we'll talk about briefly, are chemical compounds that have the same molecular formula, but they're different in the way their atoms are arranged. Molecular weight of a compound is the sum of the atomic weight of its atoms. So if you have glucose, C6H12O6, the 6 carbon atoms each weigh 12, that comes out to 72. The 12 hydrogen atoms each weigh 1, that comes out to 12. 6 oxygen atoms each weigh 16, that would be 96. So if you add 72, 12, and 96, you would get a molecular weight for glucose of 180 atomic mass units. Chemical bonds that hold those atoms together are one of two types, either an ionic bond or a covalent bond. An ionic bond is the attraction of a cation to an anion. So the sodium chloride we looked at is an example of an ionic bond. The ionic bond is relatively easy to break by something that has a more attractive power than the two ions, water being one of those things, one of the reasons water is such an important compound in life. The other type of bond, the more common type of bond that we deal with in the biological molecules, is a covalent bond. And in a covalent bond, atoms are going to share one or more pairs of electrons. So we're talking electrons, but it's pairs of electrons. In a single covalent bond, the nuclei would share one pair of electrons. In a double, they would share two pairs of electrons. If the electrons are equally shared, and depending on its geometry, it would be a non-polar covalent bond. Carbon atoms joined together form a non-polar covalent bond. There's no plus side and minus side. But if you get a molecule that, because of its geometry and because of its the attractivity between the atoms involved, you can get a polar covalent bond. And a polar covalent bond would have one side that is more negative and one side that is more positive, and that will have a role in separating ionic bonds. So here we have a hydrogen atom combining with a hydrogen atom to give you H2, and instead of calling that hydrogen compound, they would call that a hydrogen molecule because it's all hydrogen. It's not two different atoms. In a double covalent bond, there would be two sets of two shared between the carbon and the oxygen. So we would have the carbon dioxide molecule. Non-polar would be carbon-carbon bond. Polar would be the oxygen-hydrogen bond because we have a plus side and a minus side. And here we get equal sharing on both sides. One of the things that we need to talk about, which falls under the blanket of chemical bonds, is a group of forces called van der Waals forces that are weak, brief interactions between atoms. Changes in the electron density within an atom can create polarity for a moment and attract the adjacent atom for a very short time. They're only about 1% as strong as a covalent bond, but they do play an important role in physiology. They are not true chemical bonds. They're not holding compounds together, but they are used to give three-dimensional shape to compounds, especially those of proteins. The most important one of these van der Waals forces is called the hydrogen bond. Again, it's a weak attraction between slightly positive hydrogen atoms and slightly negative oxygens or nitrogens in another molecule or compound. They're relatively weak, but they are extremely important to physiology. Water molecules are attracted to each other by hydrogen bonds. Large molecules, DNA and proteins, get their three-dimensional shape in part because of the formation of hydrogen bonds within them. So they are called a bond, but again, it is not a bond that holds atoms together in a compound. It's a bond that gives you three-dimensional structure. So water molecules form hydrogen bonds because a hydrogen from one water molecule will be attracted to the negative charge of the oxygen atom. Skip the slide there. So fluids are complex mixtures of chemicals. Most of our mixtures, and a mixture is substance that keeps its physical and chemical properties, but they are mixed together. Water is going to be the basis of almost all the mixtures we talk about this year. Lots of things will dissolve in water. Water is going to end up to be about 50 to 75% of the body weight. It's because of those V-shaped, or often described as Mickey Mouse shape, of water molecules. They have properties that water has properties that make it unique in its ability to support life. Water is a great solvent. It is cohesive, it sticks to itself. It is adhesive, it sticks to other things. It's a great chemical reactive environment, and it's very thermal stable. If we look for life on other planets, we don't look for proteins or nucleic acids or nucleotides. We look for water, because even if the life on other planets is not based on carbon, as we understand it, it would need to be in a water environment. So solvency is the ability to dissolve other chemicals, and water is often referred to as a universal solvent because it dissolves many things. It is not truly a universal solvent. It will not dissolve hydrophobic substances, but metabolic reactions depend on water being solvent. Hydrophobic substances do not dissolve in water. They're nonpolar and neutral, but hydrophilic substances will dissolve in water. They're polarized or they're charged. To be soluble in water, a molecule must be polarized or charged. So attraction between the water and the sodium and the chlorine in a solution causes hydration. And as we see here, in a sodium ion, all of the oxygens are oriented towards the sodium ion, and over here, all of the hydrogens are oriented towards the chlorine ion, physically separating them into two ions. Adhesion is the tendency to stick to another substance. Water adheres to membranes, and that helps to reduce friction around the organs. Cohesion is to stick to each other. Water is very cohesive because of its hydrogen bonds. That gives you surface tension on a water surface, and that becomes very important, particularly in the lungs. Chemical reactivity is the ability to participate in chemical reactions, and water will ionize into hydrogen ions and OH ions, and it will cause other chemicals to separate and become as it did with sodium and chlorine, and it's involved in two major types of reactions: hydrolysis, which is generally the breaking of a bond, and dehydration, synthesis, which is the building of a bond between molecules. It has a high heat capacity. Calorie is the amount of temperature raised that required to get 1 gram of a substance to increase by 1 degree. So the base unit of heat, the calorie, is the amount of heat to raise the temperature of 1 gram of water by 1 degree Celsius. And this makes water very good at stabilizing internal temperatures. Hydrogen bonds resist temperature increases. It will absorb a lot of energy before it changes temperature, and it has a high heat of vaporization. So mixtures of other substances in water are either going to be solutions, colloids, or suspensions. A solution is particles called the solute, whichever is the more abundant, and almost always in physiology that will be water. And what is solute is what's dissolved in it. The solvent would be water in almost all cases. However, the solute can be a gas, a solid, or liquid. Each of those will dissolve into solvents. Solutions are defined by the following properties: a solute particle under 1 nanometer. Solute particles do not scatter light. It will pass through most membranes, and it will not separate on standing. So a glass of Kool-Aid is a good example of a solution. Colloids are often mixtures of proteins and water. They can change from liquid to a gel state, and we'll talk about that when we talk about blood clotting. Colloids have different sized particles. Their particles are generally from 1 to 100 nanometers in size. They often scatter light, usually cloudy. Their particles would not pass through a semi-permeable membrane, but the particles tend to remain permanently mixed within the solvent, even when the solvent is standing. Shaving cream and milk are good examples of colloids. A suspension, on the other hand, the particles are bigger than 100 nanometers. They're too large to penetrate selectively permeable membranes. They are cloudy or opaque in appearance, and if you leave them sitting, they will separate just from the force of gravity. Blood cells in blood plasma would be an example of a suspension, and a more common suspension you might be aware of is muddy water. An emulsion is a suspension of one liquid in another, so oil and vinegar, salad dressings, fat, and breast milk. And when we get to digestion, we'll talk about emulsifying agents. One of the issues with water, because it will separate into OH and hydrogen ions, is that of acids and bases. And acids and bases can be measured using a scale called pH. So a substance can be an acid or base depending on whether it releases or binds hydrogen ions. An acid is a proton donor, and if you think about it, a hydrogen ion is a proton. Most hydrogen is one proton surrounded by one electron. So if you have an ion, you remove that electron, you're left with a hydrogen nucleus, and the hydrogen nucleus is a proton. So don't get confused by protons and hydrogen ions being interchanged. So an acid is a proton donor. A base is a proton acceptor. It will accept that hydrogen ion, or it will release OH ions into the water. Acidity is measured by the pH scale. It's derived from the molarity of hydrogen ions. What you really need to be aware of is that a pH of 7.0 is neutral. The number of hydrogen ions is exactly equal to the number of OH ions. If the pH is less than 7, the solution is said to be acidic, and there are more hydrogen ions than OH ions. If the pH is greater than 7, the mixture is said to be basic, and there are more OH ions in the solution than there are hydrogen ions. Normal pH, which is slightly basic, is crucial for most physiological biological functions. You need to have the correct shape of proteins. Correct shape of proteins is often due to hydrogen bonds, and you can imagine that if you upset the number of hydrogen ions in a mixture, you're going to have an effect on hydrogen bonds, giving three-dimensional shape to proteins. So within the body, we have buffers. Buffers are chemicals that can resist changes in pH. To calculate pH, you take the negative logarithm of hydrogen ion molarity. You may have to calculate this in some of your homework, but what you really ought to keep in mind is that pH is a negative logarithm scale. So because it's a negative logarithm scale, low numbers give you more acidity. Higher numbers give you more basic solutions. And because it's a logarithmic scale, a change of 1 point on the pH scale represents a 10-fold change in hydrogen ion concentration. So a pH of 4 is 10 times more acidic than a pH of 5. So we have acidic substances from hydrochloric acid all the way up to pure water, which is 7. And on the other side, we have things like egg whites all the way up to oven cleaner, lye, sodium hydroxide. We've already used the term molarity, but let's talk a little bit about molarity. Molarity is the number of moles of a solute per liter of solution. A mole is like a dozen. A dozen is 12. A case is generally 24. A mole happens to be 6.02 times 10 to the 23rd. So 1 mole of the substance you have is equal to its molecular weight. So if you have 1 gram of hydrogen, turns out you happen to have 6.02 times 10 to the 23rd atoms of hydrogen. So that's 1 mole. 1 mole, think of it, don't get tied up in the 6.02 times 10 to the 23rd. Just think of that mole as a specific number, like a dozen, like a case. Often, it's the most physiological, meaningful measure of a concentration. Body fluids are usually quantified in millimolar concentrations. Milliequivalents are similar, except they have to do with the electrolyte concentration, the millimolar concentration of a solute, and the electrical charges of its particles. In many cases, the milliequivalents and the mole and equivalents are the same, but if you have a divalent ion, for example, it will be different. But it turns out that those are very important for nerve firing, heart beating, muscle contractions, and delivery of intravenous fluids. So you do have to be aware of them. Now a little bit of physics, since we're going right along. Energy is the capacity to do work. To do work means to move something, a muscle or a molecule. Potential energy is energy stored in an object but not currently doing work. It could be chemical energy or free energy. Kinetic energy is the energy of motion or energy doing work. Muscle movements, flow of ions, vibrating eardrum. Heat is kinetic energy of molecular motion. Electromagnetic energy is the kinetic energy of photons. Electrical energy has both potential and kinetic forms. And those will be important as we go out the year. So let's talk about how those atoms come together in chemical reactions. A chemical reaction symbolizes the course of a chemical reaction. You write them with the reactants on the left, an arrow, and the products on the right. There are some common classes of reactions that we'll go through quickly. Decomposition, synthesis, and exchange reactions are all important. In a decomposition reaction, large molecules break down into two or more smaller molecules, so the generalized formula AB yields A plus B. In a synthesis reaction, two or more small molecules combine to form a larger one, so A plus B yields AB. In an exchange reaction, molecules exchange atoms or groups of atoms, so AB plus CD yields AC plus BD. An example of that would be hydrochloric acid and sodium bicarbonate exchanging hydrogen ions to give you sodium chloride and bicarbonate. Most reactions are reversible. We draw the arrow in one direction, but depending on concentrations, they will go in both directions. So you should really be drawing the double-headed arrow. This is very important in reactions in things like respiratory biochemistry, urinary biochemistry, digestive physiology. Reversible reactions will follow the law of mass action. So you can, at equilibrium, they would be stable, but if you had more product than reactants, that will break down into reactants. Reactant rates reactant rates are affected by things that will affect the amount of energy or the amount of times that molecules collide with each other. And so things that will increase the rate of reaction are things like concentration, temperature, and a catalyst. Catalysts we'll talk about in a moment, but they are going to speed up chemical reactions by lowering activation energy. They are not changed in the reaction, but they help the reactants get into the correct shape to have a good collision. All of these chemical reactions are collectively known as metabolism. And metabolism can be broken down into two types of reaction. Catabolism, which is an energy-releasing decomposition reaction, breaks covalent bonds and produces smaller molecules. The other side of the coin is anabolism, energy-storing synthesis reactions. They require energy input, and they're going to produce things like proteins and fats. So catabolism and anabolism are inseparably linked. The energy for anabolism comes from catabolism. Some typical reactions might include oxidation, in which a molecule gives up electrons and releases energy. So the molecule is oxidized. When it loses electrons, the oxidizing agent is the electron acceptor, and that is often oxygen. And a reduction reaction is a chemical reaction in which a molecule gains electrons and therefore gains energy. Molecule is reduced when it accepts those electrons, and the reducing agent is the molecule that donated the electrons. Oxidation of one molecule is always accompanied by the reduction of another, and we get an oxidation reduction or redox type of reactions. And again, this has to do with why reactions can occur, because the energy for one comes from the other. Redox reactions are very important in living science, living physiology. All right, now we're going to start to get into the 4 types of molecules that are going to be most important to us all year. These are organic compounds, and they are polymers of organic compounds. The 4 big ones are carbohydrates, lipids, proteins, and nucleic acids. Carbon works because carbon has 4 valence electrons, can form 4 bonds with up to 4 different atoms. It readily combines with other carbon atoms to form carbon backbones, which can be chains, branched, or rings. It combines well with oxygen, hydrogen, nitrogen, and sulfur, and several other elements. Those carbon backbones can have a variety of functional groups, the small clusters of atoms attached to the carbon backbone, and that will determine many of the properties of the organic molecules, things like hydroxyl, methyl, carboxyl, amino, and phosphate. Carbon is very good at making macromolecules, which are large organic molecules that have high molecular weight because they're large. And most of the macromolecules that we deal with are polymers. They are long chains of repeating subunits. We call them monomers. Monomers could be identical or they could be different. Starch is a polymer of about 3,000 identical glucose monomer, and DNA is a polymer of 4 different nucleotide monomers. Polymers are formed by polymerization, which is the chemical joining of 2 monomers. Monomers link together by dehydration synthesis or condensation reactions, in which a hydroxyl group is removed and a hydrogen from another, and then you get a water molecule from that, HOH. The opposite of that is hydrolysis, and then water molecule is used to split the 2 polymers, and OH goes to one and H goes to the other. So monomer 1 forms through a dehydration synthesis, and later on that dimer can be split by adding the water back into a monomer 1 and monomer 2, what you had before. So the first of these compounds is going to be carbohydrates. Carbohydrates are hydrophilic organic molecules. Their major role in the cell is for energy. They have the general formula of CH2O to the N, where N is the number of carbons. So glucose would be C6H12O6. There's always going to be a 2-to-1 ratio of hydrogen to oxygen. And if you think about it, H2O is water, and these are really hydrated carbons. Names on the carbohydrates are often built from the root saccharide and the suffix "-ose, both which mean sweet and sugar. So sugars and starches are carbohydrates. Simplest carbohydrates are the monosaccharides, and there are 3 important monosaccharides in physiology: glucose, galactose, and fructose. They're produced by the digestion of more complex carbohydrates. Glucose is blood sugar. All 3 of them have the same formula, C6H12O6, but this is an example of those isomers that I mentioned earlier in the lecture. Ribose and deoxyribose are also monomers, they are part of DNA and RNA. So here's glucose, galactose, and fructose. When you cook 2 monosaccharides together, you get disaccharides, and these disaccharides are important for our diet supply of monosaccharides. Sucrose is a glucose and fructose, that would be table sugar. Lactose in milk is a glucose and a galactose, and maltose, which is in grain products, is a glucose and a glucose. Oligosaccharides would be short chains, 3 or more monosaccharides, but from disaccharides, we generally go to polysaccharides, which are long chains of monosaccharides, 50 or more, up to thousands. There are three important polysaccharides for us: glycogen, which is the energy storage molecule in the liver; starch, which is a similar molecule but made in plants, but it is digestible by humans; and cellulose is also made in plants. It's important. We call it fiber in our diet, but we cannot digest it, but it's still important. So here's glycogen with lots of branches. Carbohydrates mobilize quickly to give us energy through the oxidative phosphorylation pathway. They are often bound to lipids and proteins, so we have glycolipids and glycoproteins. They form structures called proteoglycans that are more carbohydrate than protein, which forms some gels that hold tissue together, fills the umbilical cord in the eye, and lubricate joints. And they have a moiety for each of the components of a conjugated macromolecule. Lipids, the second group, are hydrophobic organic molecules with mostly hydrogen and carbon, very little oxygen. They have more calories per gram than carbohydrates, and there are 5 primary types that we'll look at: fatty acids, triglycerides, phospholipids, eicosanoids, and steroids. Fatty acids are chains of 4 to 24 carbon atoms with a carboxy group at one end and a methyl group at the other. There are some that are essential, which means they must be obtained from your diet. We can't make them on our own. Fatty acids are classified as either saturated or unsaturated. A saturated fatty acid, all the carbon atoms are linked by a single covalent bond. They're saturated with hydrogens, is where they get the name. Unsaturated fatty acids contain double bonds between the carbons, and the molecule still has the potential to add more hydrogens. If there's more than one double bond, then it is a polyunsaturated fatty acid. Triglycerides are 3 fatty acids that are linked to a short carbon chain called glycerol. It has three carbons in it. They're formed by dehydration synthesis, and they're broken down by hydrolysis. Their primary function is to store energy and to help with insulation and act as a shock absorber. They are also called neutral fatty acids because once they're formed, the fatty acid is no longer acidic. Dietary oils and fats are typically triglycerides. If they are oils, they are liquid at room temperature. If they are fats, they are solid at room temperature. So here is your glycerol molecule and 3 fatty acids attached to it, and these are saturated fatty acids. This is a unsaturated fatty acid. They can also be cis or trans. If you have a double bond, it can be relatively straight or it can have a kink in it. And if it's relatively straight, it is a trans fatty acid. If it's got a kink in it, it's a cis fatty acid. Phospholipids are similar to triglycerides, but one of those fatty acids is replaced by a phosphate group. And that phosphate group makes this molecule amphiphatic. They have a hydrophobic region and a hydrophilic region, a part that likes to be in water and a part that doesn't like to be in water. Phosphate groups can also be linked to other functional groups, and they are the structural foundation of cell membranes. So we have a hydrophilic phosphate group at one end and 2 hydrophobic tails at the other. Eicosanoids are 20 carbon compounds derived from a fatty acid called arachidonic acid. They act as hormone-like signals between cells. They would include the prostaglandins, and prostaglandins have a very large role in inflammation, blood clotting, hormone action, labor contractions, and blood vessel diameter. Steroids are 17 carbon atoms in 4 rings. Cholesterol is what all the steroids are derived from. It's important for the nervous system function and for the structural integrity of cell membranes. We get about 15% of our cholesterol from diet, but about 85% of that is internally synthesized, and it's done so in the liver. Other steroids would include cortisol, progesterone, estrogen, testosterone, and bile acids. Here's that cholesterol molecule, which is the basis for all of those steroids. You've probably heard of good cholesterol and bad cholesterol. Well, turns out this is the only cholesterol. When you hear good and bad, they're referring to cholesterol being carried through the body. HDL, or high-density lipoproteins, are what's typically referred to as good cholesterol. They have a lower ratio of lipid to protein, and they may help prevent cardiovascular disease. LDLs, or low-density lipoproteins, are typically referred to as bad cholesterol. They have a higher ratio of lipid to protein, and they are known to contribute to cardiovascular disease. Bringing us to the proteins. Protein is a polymer of amino acids. Amino acids have a central carbon with 3 attachments. There is a amino group, a carboxy group, and an R group. There are 20 possible R groups, and the properties of each amino acid is basically determined by the properties of that R group. Amino acids join together with a bond known as a peptide bond. A peptide bond is going to join a carboxy group of one to an amino group of another, and it will be a dehydration synthesis. Peptides are named for the number of amino acids they contain, so a dipeptide would be 2 amino acids, tripeptide 3. Oligopeptides are between 10 and 15, and bigger than 15 are polypeptides, and if we know what they do, we call those polypeptides proteins. So we have amino acid 1, amino acid 2, an OH from this amino acid, and an H from this amino acid forms a bond between this carbon of the amino group and this, of the carboxyl group, and this nitrogen of the amino group. And you're left with water because it's a dehydration synthesis. And this carbon-nitrogen bond is called a peptide bond. So 2 amino acids, this would be a dipeptide. Proteins have a very complex 3-dimensional structure that we call their conformation. That conformation is unique, and it is crucial to the way that protein functions. Proteins can reversibly change conformation to affect function. We see that in muscle contraction, enzyme catalysis, membrane channel opening. We'll see it in the eye when we talk about how vision occurs. Denaturation is an extreme conformational change that destroys the function. Things like extreme heat or pH can cause denaturization, and that would be permanent in those cases, irreversible. A cooked egg white is denatured and becomes an opaque, stiff molecule structure instead of a fluidy structure. So to get to that final conformation, we describe proteins as having 3 or 4 levels of complexity. The first one is the primary structure. That is the sequence of amino acids within a protein molecule. That's determined by the genes. Those amino acids will then coil and fold up because of hydrogen bonds into short regions of structure called the secondary structure. So the secondary structure of a protein may have examples of alpha helix, beta sheets, pleated sheets. The total conformation of that single protein molecule is known as its tertiary structure. That is the 3-dimensional shape that you get by folding and bending the secondary structure and a primary structure into its final three-dimensional shape. And that may include the formation of disulfide bonds if you have the amino acid cysteine in there. That tertiary structure is generally either a globular protein, which is compact, found within cell membranes, move freely in the body fluids, or fibrous proteins, which tend to be filaments, and they have a lot of structural function within the body. Now, if two proteins come together to give you a structure, then we have to add the quaternary structure. And quaternary structure involves the final three-dimensional shape of all the proteins involved in making that substance. For example, hemoglobin has 4 peptide subunits, so it has quaternary structure. Primary structure, amino acid sequence. Secondary structures form within those amino acid sequence, and then they fold together to give you tertiary structure. And if you only have one polypeptide making that protein, that's all you need to worry about. But in many cases, we have that polypeptide working with other polypeptides to give you your functional protein, and you would have quaternary structure. Those proteins are going to serve structural functions. Keratin is one we will talk about very soon when we talk about the integumentary system. Collagen is a very abundant protein, deep layers of the skin, bones, cartilage, and teeth. Proteins serve communication functions, neurotransmitters, hormones, and other signaling molecules. Those signaling molecules that bind to something else are called ligands. And the receptors would bind to those ligands. Those are proteins as well. They're involved in membrane transport, getting things from one side of the membrane to the other. Many of them are enzymes that speed up chemical reactions, so they do catalysis. And many of them are used for recognition and protection. They're used to move things. We have motor proteins, and they are used to hold cells together, cell adhesion proteins. So those enzymes that serve as catalysts, and they're not all proteins. We do have some that are made of RNA. They are ribozymes. But an enzyme acts on a substrate. It speeds up a chemical reaction by lowering activation energy. And that activation energy is the amount of energy you need to get a reaction started. So they permit reactions to occur at body temperature. They're named by using ase as a suffix very often. So amylase is going to hydrolyze amylose, a starch. Lactase is going to catalyze the breakdown of the disaccharide lactose. So the first thing that has to happen is the substrate binds to a pocket on the enzyme called an active site. That formation of enzyme substrate complex is thought to be like a lock and key. Enzyme would then release the reactant products. The enzyme remains unchanged. So sucrose is hydrolyzed by sucrase to produce glucose and fructose. So again, the enzyme binds to the reactant. The shape of the reactant is changed into the products, and the enzyme releases the product. So substrate binds to the active site, a reaction occurs, and the products are released. Temperature, pH, and other factors can change the enzyme shape and therefore affect its function. They can alter the ability of the enzyme to bind its substrate. Enzymes have an optimum pH. Most would be around physiological pH, but pepsin in the stomach works best at pH of 2. And the temperature optimum for human enzymes is also usually near 37 degrees Celsius. Enzymes also use non-protein partners called cofactors. They can be inorganic or organic. Organic cofactors are often called coenzymes. We'll see NAD later on when we talk about oxidative phosphorylation. Those enzymes are involved in metabolic pathways, which are a chain of reaction. Each step in the chain is catalyzed by a different enzyme. So here we have alpha, beta, gamma. Alpha is the enzyme to change A to B, beta changes B to C, and gamma changes C to D. Those metabolic pathways are turned on and off by affecting the enzyme activity. That could be the binding or dissociation of cofactors. It could be the buildup of products. And it could be turned on and off by genetic means. Last of them is the nucleotides. Nucleotides are organic compounds with a nitrogen base, a sugar, and one or more phosphate groups. ATP is an adenine with a nitrogenous base, a ribose sugar, and 3 phosphate groups. We're going to talk a lot about ATP this year. There's ATP. It's the most important energy transfer molecule. It stores energy gained from exergonic reaction, releases it within seconds for physiological work, holds the energy in covalent bonds between the phosphates. Hydrolysis of ATP is catalyzed by adenosine triphosphates, ATPases. It breaks the third energy bond to produce ADP, and then it can be phosphorylated to build ATP again. That's carried out by an enzyme called a kinase. Energy for ATP comes generally from glucose. The first stage would be glycolysis, in which the glucose is split into 2 pyruvates. Pyruvates, if there is oxygen present, will go through aerobic respiration in the mitochondria. If there's not, then it will go into anaerobic fermentation or lactic acid fermentation, as you may have heard it called in other classes. The other nucleotides are guanosine triphosphates, which are similar to ATP, but they are used in some reactions. Cyclic AMP is another important compound that is a second messenger to in many reactions. And the final 2 nucleic acids are the ones you're probably most familiar with. DNA, deoxyribonucleic acid, which makes up the genes, the instructions for synthesizing proteins, and RNA, or ribonucleic acid, which takes those instructions and uses them to build the proteins. All right, I want to thank you for listening. This is a long one, probably the longest one you will have all year, and there is a lot of information in chemistry, but you will get through it.